Okay, thank you very much for the introduction, and I'd like to thank the OTS for the honour of this award and for the opportunity to talk to you all today. Okay, so I'm interested in respiratory viruses, and that's because seasonal respiratory infections have been estimated to kill almost 4 million people every year. Now, respiratory viruses normally cause acute infection by infecting the epithelial cells of the upper respiratory tract. And the two viruses I've got here, influenza virus and respiratory syncytial virus, or RSV, are two of the lead causes of respiratory viral pathogenesis, causing significant morbidity and mortality. Now, for both these viruses, and really the majority of acute respiratory viral infection, there's an unmet clinical need. For RSV, there's currently no post-exposure therapeutic. And for influenza virus, we're seeing the emergence of multi-drug resistant strains. And that goes back to the central dogma of antiviral therapeutics, which is predominantly, the discovery has been against direct targeting antiviral therapeutics. But over time, viruses have the ability to resist and really make these therapeutics not work anymore. The second method that you can use to create novel antiviral therapeutics is by targeting host factors that are essential for virus replication. And that's the method that we've gone through, mainly because the Buck lab discovered in 2010 microRNA mimics with broad-spectrum antiviral properties. And this was done through a mimic inhibitor microRNA library against GFP reporter viruses of the herpes virus family. And this identified 20 microRNAs that had broad-spectrum effects against the alpha, beta, and gamma virus herpes viruses. And then we further extended this into the small RNA virus, Semliki forest virus, and noticed that some of these microRNAs that were active against these large DNA viruses were also active against this small RNA virus. And that made us think, well, it's probably not targeting the viruses directly, and much more likely to be targeting host factors that these viruses have in common. We, of course, wanted to extend that into respiratory viral infection. So I've just got here a fairly simple schematic about the testing we did with influenza and RSV. So this was a simple reverse transfection and subsequent infection with, on the left, influenza H1N1 WSN strain, and on the right, RSV A2. And then we have a look at the effects on viral titer from each of our microRNA treatments. I'm not going to go through these graphs in detail. The negative controls are in black, and then we have our various microRNA treatments in white. And that's because I've summarized it all in this handy table for you. On the left, we have the previously published broad-spectrum activity against the herpes viruses. And then we have further extended, as I mentioned, we've tested multiple strains of influenza H1N1. We've also tested it against influenza H3N2, so both A strains of influenza virus. We've tested against the laboratory strain, so RSV A2, and we've recently got our hands on a clinical strains of RSV, and we've done some testing of our 20 microRNAs against this as well. So all the ticks refer to microRNAs with at least a half a log or a 50% decrease in viral titer with fairly minimal effects on cell viability. And we wanted to pick lead candidates from this, and quite quickly we were able to identify 4 microRNAs. That is MER542, MER744, MER24, and MER124A that had broad-spectrum antiviral properties against all the respiratory viruses that we were testing against. Now, of course, the thing that we're all asking ourselves is what could potentially be the mechanism of action of these antiviral microRNAs? And to get a really broad idea about what might be happening in the cell, we chose to do a reverse phase protein array as one of the arrays that we did. This was a similar protocol to what I've already said, with a reverse transfection, subsequent infection with influenza H1N1 WSN strain, and then taking the protein lysates for this RPPA. We then dot it out on a microarray substrate and probe with primary antibodies to we chose global pathway regulators, and we also chose factors that have been previously associated with influenza virus infection. With a secondary that is fluorescent, we can then have a look at the relative abundance of all these proteins and have a look at some pathway profiling. I only have time, unfortunately, to show you just one of these that came up as interesting as part of this screen, which was MAP-Cap kinase 2. And it's involved in multiple different pathways and is actually activated via stress, one of those stresses being influenza virus infection. And when we compare it to, so this is just an infected control, we have a risk-free siRNA there in blue as our negative controls. MIR542 shows no activity in white, but the other 3 microRNAs that were part of our lead candidate portfolio, MIR744, 124A, and 24, are all causing the suppression in MAP-Cap kinase 2. So we know that this P38 MAP kinase pathway is important for viral yields and the nuclear export of viral RNPs. And we also know that the reduction in MAP-Cap kinase 2 actually causes PKR activation and the downstream antiviral effects that PKR has. So we just wanted to validate this. As I said, this was just an initial look. So we basically repeated the assay with different concentrations, time points, threw in some more controls. And this is just an example of a Western blot with MAP-Cap kinase 2 being these bottom 2 bands at the bottom here. And this is quantified for you on the right, where you can see again that compared to the controls on this side, the mimics of 744, 124A, and 24 are all causing the potent suppression of MAP-Cap kinase 2. Whereas the inhibitors, at least for 124A and 24, we're seeing that differential response, which is nice to see. So we're expecting this suppression of MAP-Cap kinase 2 to be responsible for some of the antiviral effects that we may be seeing. But we want to know, well, what is actually the microRNAs targeting? Because MAP-Cap kinase 2 is not a direct target of any of these microRNAs. And to do this, it's, as we've been previously heard throughout the conference, it's to do with this seed region of microRNAs and the fact that they can target at multiple different mRNAs. So we can use prediction algorithms, but then you end up, because there's other factors involved, with false positives that you may be chasing after. We tend to instead concentrate on what's already been experimentally validated for our microRNAs as sort of a first step to having a look at what might be their targets. And this is an example for MIR24 that I've put for you on the right. And I've just got MAP-Cap kinase 2 here in a box. And I've inserted that into something that, a string diagram of the protein interactions of all the experimentally validated targets of MIR24. And you can see here, if I just enlarge that, is that one of the targets of both MIR24 and MIR124A is actually this MAP kinase 14. And that's the P38 alpha MAP kinase that's known to activate MAP-Cap kinase 2. So we can see that these microRNAs are affecting multiple points in that MAP kinase pathway. And as I said, this is just one example for you today of a target that's been validated in influenza virus infection. But what's really exciting about this work is that we can use microRNAs as probes and identify new networks and pathways that are important in respiratory viral infection. Okay, so we have our microRNAs that are causing antiviral effects in vitro. Do they actually work in vivo? To have a look at that, we needed to deliver our microRNA mimics into the lungs of mice. And we've chosen animal models of respiratory viral infection. So in total, the project's tested 10 different delivery vectors, and these have a range of different properties, a range of different sizes, and we've tested both intranasal and inhalation with the majority of them. And I can tell you that with inhalation, we really do it's not only more clinically relevant, but we get more uniform delivery and we get much less immune activation with this delivery, as you'd expect. The delivery vector that's been studied most extensively within the lab is EPEI, which is a cationic branch polymer that the Cystic Fibrosis Consortium studied extensively during their development of a gene therapy. And in our hands, when we have a look at aerosol delivery, our preliminary tox has shown that we're not seeing any significant cytokine production or immune cell influx. And when we have a look at delivery, so this is just an example here for you on the right. So a single delivery of the microRNA mimic MIR542 via the nebulization route. And then we have a look at 48 hours later, and this is just looking at microRNA copy number. In your untreated mice, this is the normal levels of MIR542 within the lungs. And then with that single treatment, we get a 10 to 100-fold increase in total lung levels. When we look at the bowel, it's very similar. And then the lab has also designed a new method of digesting the inner epithelial lining from the lungs. And within that inner epithelial lining, we're also seeing the 10 to 100-fold microRNA increase. And these are then the cells that are infected and that we're mainly interested in. So we get delivery. Does this actually result in an antiviral effect? So I'll just go through our prophylactic model, which is administering our microRNA with the EPEI via nebulization, a day later subsequently infecting the mice, and then 2 days later having a look at viral titer in total lung via plaque assay. And compared to a naive mouse, or this time we've used C. elegans control microRNA, MIR542 treatment has caused a significant decrease in influenza virus infection within the lung. And look, we've also done a therapeutic regime, and that's just infecting mice first, and then this time giving 2 doses of our microRNA mimic. And again, when we have a look at total virus load, we're seeing that significant decrease from MIR542 treatment. And I'll just refer you back to the prophylactic treatment because this is the model that we've used most extensively. And while we do see a significant decrease, this has only been from 2 to 10-fold. So the maximum we've been able to achieve is a log decrease within the lung with that prophylactic model. And I really think that that's the sheer fact that we just don't have delivery sorted yet. And we could definitely optimize within this area. Okay, so in some conclusions, we have these broad spectrum microRNAs that we've screened for activity against respiratory viruses, and we've chosen 4 lead candidates. We're continuing to test and broaden this range of viruses that have activity, so currently testing against rhinovirus, and we're also testing with some pathogenic viruses in collaboration with the US Army. We've done some mechanism of action studies, and that's given us some really interesting results on possible antiviral pathways that our microRNA mimics are working in. But we're currently undertaking some further analysis, and we want to really pinpoint what are our direct microRNA targets. And finally, I've shown you some in vivo studies which demonstrate that our microRNA mimics do have antiviral potential, but that we really want to have a look at maybe some novel inhalation delivery vehicles or further optimization of our delivery. And I'll just finish with acknowledging everyone who's been involved in the project. So my mentor and supervisor, Amy Buck, who's really the main driver of this project and has been phenomenal. The rest of the group in the Buck laboratory, it's been a really University of Edinburgh focused project. So members out at the Roslin Institute have been involved with delivery and some of the animal models of infection, our RSV experts and RPPA collaboration, US AMRID that I mentioned that we're doing the pathogenic viruses with, our funders, and finally OTS and the sponsors of the Memorial Scholarship. And thank you to you all.
Thank you, Jana.
Thanks.
Great talk. Yes, Rachel.
So 2 questions. The first one is, in the in vivo delivery experiments, do you actually need the PEI? Have you done the experiments without
We have. Yeah, so we've done those experiments and we see very minimal delivery with the nebulization.
This is nebulizing in mice?
Yeah, so we don't do the most straightforward method in the fact that we actually nebulize a whole cage. So you can have one mouse in there, you can have 40 mice in there, and we just nebulize that entire cage per group. What we're nebulizing, we're getting so much waste. And one of the things that we want to move towards is, getting that nose cone applicator. And we were meant to be getting that 6 months ago. It hasn't happened. So we've had that delay, but definitely something that we want to try.
My second question was, and you may have said this and I missed it, the relationship between those 4 different microRNAs with respect to targeting their seed sequences and what you think might be commonalities, pathways that they're engaging. What do you know about that?
Yeah, look, it is work in progress as far as that. They are fairly distinct microRNAs. So seed region, they don't have commonalities. What we're noticing when we're doing these analyses, so both at a protein and an RNA level, is some of the microRNAs are targeting similar pathways, and then we're seeing obviously the differences in them. And I think it just showed us really the power of the microRNA in the fact that we get one microRNA that's actually hitting multiple different antiviral pathways. And that's one of the reasons why we're excited about this work.
Yes, Art.
I was just wondering, the C. elegans control RNA that you used,
Yep.
how similar is that to your microRNA? And were there other control RNAs that you used also?
Yeah, so we do try and use a couple of negative controls just because with any sort of microRNA mimic, you're going to see off-target effects. So your C. elegans negative control has no sequence similarities to any of these microRNAs, but it will be having off-target effects with the nature it is an RNA that we're putting in. So we also tend to use microRNA MR30D that we've shown isn't effective in influenza virus infection. Actually, the majority of respiratory viral infections, it has no antiviral activity. So of course, still having those off targets, but we're not seeing them at an antiviral level.
Okay, if there are no further questions, remains for me to present this on behalf of the OTS and congratulate you again for your award.
Wow, thank you. Thanks so much.